Okay, it's 5 o'clock. I've got 64 of you with me so far, which is quite a long way short of the total number, but since this is being recorded, hopefully people can catch up with it when they want to. Glad to hear that other people are checking the US election results too. I'll try not to be too political. I have a bit of a tendency to go on political rants in my lectures, so I'll try to be a good boy, but I apologise in advance if I do go off on one. So welcome to the Experimental Design and Analysis section of the Core Skills course. So this is a set of content about the fundamental building blocks of experimental design, and then quite a lot of information about statistical analysis, which is relevant to all of the degrees in life sciences. So everybody apart from the pharmacology students should be joining in at some point. And the first session I decided to have live just because it kind of felt like a nice thing to do, although the degree of interaction we can have is perhaps less than ideal. Somebody's pointing out that some people are in a calculation workshop. Yeah, I was just talking to Tim Simpson about that. We realised there was a clash that he hadn't figured was there. So that's okay. We can catch up. You can catch up in due course with the recording. Yeah. So the sessions for this part of the course, they're going to run between now and Christmas, and then beyond Christmas until about halfway through the next semester. And there'll be 1 hour a week on average of content. Occasionally there'll be 2 hours, and it'll be pre-recorded, but then every 3 hours or so, every 3 weeks as it were, we'll have a live wrap-up session where I won't deliver any new content. Instead we'll do a sort of a catch-up. You can ask questions, and I'll probably set you a Kahoot quiz or something like that to try and make it a bit more interactive, because I know how mind-numbing it is sitting at home watching pre-recorded lectures. We're learning that from you, particularly in first year. I think it's a big challenge for you guys. So this part of the course is really about how to do science. It's about how we go from having a cool scientific idea to getting an answer to the question that we are posing. And so it's my job to try to teach you some of the rules of engagement for that process. And in this first lecture, we're really going to look at how the scientific process works. If I can get to my content slide, here we go. So before I do that, I'm going to give you a bit of an outline for this part of the course, some of the rules, some of the things that are going to happen to you, which includes, of course, assessment. So I will be covering the exam briefly and mentioning the coursework that you'll have to do, which will vary depending on which degree group you're in. And then I'm going to move on to look at this scientific process, or what might be called the scientific method. So a key learning outcome from today is for you to be able to describe the scientific method in its sort of fairly simplified form that I'm going to give you today. And that will bring us to a key concept in experimental design, which is the hypothesis. So we're going to look at what a hypothesis is and how to make a good hypothesis, because that's often the starting point for doing good science. And in particular, we're going to look at the idea of a null hypothesis and its opposite, which is the alternative hypothesis. And that will bring us right at the end of the lecture to why statistics are relevant in this context, why we end up having to do statistics in order to make sense of biological data, which unfortunately is just kind of part of the job of being a scientist. Okay, so the objectives of the course are to understand the following things. First of all, as I've already mentioned, something about how the scientific method works. In other words, how scientists generally go about creating new knowledge, right from the initial part of the process, making observations and asking questions, right through to doing analysis and answering those questions. And it's going to centre on particularly formulating and testing hypotheses. Then we're going to look at the basics of experimental design, some of the things that you'll need to know in order to do a good experiment or make good scientific observations, which will ultimately give you an answer to the question that you're asking. And then for a big chunk of the course, from sort of about the fourth lecture onwards, we're going to look at different types of statistical tests that can be used to analyse data coming from different types of experimental design. A little bit about the assumptions those tests make, how they work, what they're for. And then in your own degree groups at various stages, you'll probably be putting some of these ideas into practice in different ways in different degrees, as I'll mention in a second. So one of the kind of key goals here is for you to get to a point, not necessarily where you're an expert in doing your own analysis, although you will have a grounding in that, but really so that you can understand other people's analysis when you read it, because in second and third year in particular, but all the way through your degree, you're going to be reading other people's science in textbooks, in papers. You're going to go to lectures where people are describing the results of experiments and studies that have been done. And in order to have a sort of healthy degree of skepticism and a good grasp of what results can and can't tell you, then understanding the basics of experimental design and analysis are going to be really important for you, whether you be a biochemist or a zoologist, everybody in between. So for the biology students, I'm going to be running a separate set of online workshops and activities where they have to do some coursework to put the ideas from these lectures into practice. And there are going to be similar activities going on in each of the different degree groups. So I've listed here roughly what's going to be going on. Well, specifically what kind of software you're going to be using, because in order to put the ideas, the statistical ideas I'm going to be talking about into practice, you're going to use some statistical software. So for the biology students, you're going to use a software package called R, which is freely available, and it's kind of becoming the industry standard across the life sciences. But as you'll find out, if you're a different kind of student, like a neuroscientist, for example, in different fields within the life sciences, there are different traditions and different software packages that tend to get used. So for the neuroscientist, you'll be using PRISM, and Paul Smith will be helping you to understand how to use PRISM to tackle essentially the self-same set of analyses that the biologists will use R for. The biochemist will be doing a little bit less by way of sort of practical stats with data, but you will be doing some stuff with Tim Simpson. I believe you're mostly going to use Excel, which actually does many of the things that I do in R and that the neuroscientists do in PRISM. And then finally, the sports and exercise science students use yet another software package called SPSS. And I think, as I understand it, you probably won't be doing that until you get to your third year. And the reason you're going to be doing it in third year, and the biochemists and neuroscientists and biologists will also be doing some stats in third year, is because you'll hit your project. And in your project, you're going to generate data. You're going to use that data to answer a question. And in order to do that, you're going to use statistical software and the ideas that I've introduced to you in this course. So the ultimate objective here is to give you some tools, both for designing and analysing your own studies and understanding, criticising, appreciating the value of other people's work. So this is kind of partly about stats, and so there's a kind of instinctive response from some students to think, I hate maths, or I haven't done maths, therefore what am I going to do? Am I going to cope? Or if you've done A-level maths or even A-level statistics, you might be thinking, this is just going to be boring recap of stuff I already know. Well, the thing to reassure you is that this is not a maths course. We're not going to do a whole load of maths. I am going to flash up some equations from time to time, but the emphasis is really on understanding the concepts in the equations, what the equation is for. They're not going to be, we're not going to spend lots of time rearranging equations or doing maths. I'm not going to set you maths problems to solve, for example, and you won't have that in the exam. The focus is very much on the biological context here. It's as biologists, as scientists, we are posing scientific questions, using data to answer those questions, and we're going to use statistics as a tool in order to do that. So even if you know the ins and outs of the tests that I'm going to talk about later in the course, which some of you who've done A-level statistics will, you'll see that we're learning new things in a way, because we're learning about the application of those tests and approaches rather than worrying about how the formulae are derived or what the mathematical properties of those tests are. So we're looking at relatively few equations, and we all have different backgrounds, so some of you will know some of this stuff already, others of you won't, and the idea is to bring you up to a common standard so that you all have a decent base to work from in future. And certainly in the case of the biology students, I'll be running help sessions where I can give you more focused advice if you're struggling particularly with the practical aspect of applying the ideas that I teach here, and there'll be equivalent sessions for neuroscience and biochemistry and sports and exercise science. Okay, assessment wise, you're going to do an exam which is going to be in the form of a ROGO multiple choice style exam, which will happen in May-June. And at the moment there are some uncertainties about exactly how we're going to run those exams because of COVID, but it'll basically be on the computer, probably in some kind of open book form rather than turning up to a computer room, which is what it would be in a normal year. It'll be in May or June. Oh, I've said 2020 here. That's a mistake. It's 2021, isn't it, by the time you do the exam? And it'll be worth 25% of your core skills module, unless you're a biochemist, in which case it's 34%, or a sports and exercise science person, where it's a little bit different. So let me just break that down. So for neuroscience, biochemistry and biology, you've got a separate exam, which is worth 25% or 34%, depending on who you are, of your module. For sports and exercise science students, you're going to get very similar questions, or some of the same questions, but they're going to be part of a bigger exam which also assesses other things. So slightly different format, but the same things being tested. And basically I'm testing you on the knowledge of the material that I teach in the next 10 lectures or so. And there will be a chance to do a practice. I'll give you basically a mock exam that will be made available later in the course. So don't panic about not really knowing how you're going to be assessed. Yeah, so in addition to this, many of you will be doing some kind of assessed coursework as part of core skills, which will use some of the expertise that you've gained in my lectures in this section of the course. So for the biology students, you're going to be doing a workbook which contains mini practicals where you do analysis in the software R using the ideas that I've taught you in these lectures. And there's something similar for neuroscience students using PRISM, and there's something similar for the other students as well, although it all varies a little bit depending on your degree. If in doubt, go to your convener for the relevant core skills module, so Tim Simpson if you're a biochemist, for example, and also remember to look at your core skills Moodle page for your degree, because this core skills module is complicated. We have a generic page where the material, like what I'm teaching you today, which is relevant to everybody, will be placed, but also, probably more importantly, you've got a degree specific page which takes you through week by week what you need to do if you're a biochemist or a neuroscientist or a sports and exercise scientist. So please make sure you've got the hang of those two Moodle sites and use this degree specific one as your sort of main point of entry, and it will refer you to the generic page when you need to go there. Phew. Okay, before I get stuck into the content then, the last little bit of admin is just to mention about reading. I don't expect you to go away and do a vast amount of reading to support this course. The lectures should be sort of reasonably standalone. But there is one book by my dear colleague Francis Gilbert and my dear former colleague Chris Barnard, who sadly died some years ago, and another former Nottingham academic called Pete McGregor, and they wrote this book called Asking Questions in Biology, which is a really good introduction to everything that I'm going to be talking about on this course, right from how to formulate a hypothesis through to how to use software like R and SPSS to test data sets. So that book is widely available in the library. I think we have access to the electronic copy via the library. You can go out and buy it if you want, but I try not to encourage you to line the pockets of my colleagues. So do get access to it through the library if you need, and it provides a sort of good support for what I say, but it teaches the same ideas in slightly different ways, so you may find that helpful. And then there's a couple of other books I'm recommending to help support things. So Choosing and Using Statistics by Calvin Dytham is a helpful sort of book on just the practice of doing stats in a computer using the information that I'm teaching in this course. So its focus is much less on the theory, much more on the practice of which test should I do and how do I do it. And Ruxton and Colgrave covers something rather different. It covers the do's and don'ts of experimental design. So it's not about stats, it's about how to do good scientific experiments and studies. Calvin Dytham's book, I should note by the way, doesn't deal with PRISM, so it's not going to be hugely helpful for neuroscience students. And similarly, actually, the Barnard et al. Book, the theoretical content in there is going to be relevant to everybody, but it does focus on using SPSS and R, so it's going to be more useful for sport and exercise science and biology students in that bit anyway than others. And finally, for biology degree students, I would recommend this very simple introduction to R by Beckerman and Pecci, who are academics at Sheffield, which is a good way, a helpful introduction to get started with R, especially if you find you're struggling with that. Okay, so let's get on with the actual content of this course, and I really want to start with the basics about what it is that scientists do, because I think, you know, you're going to become a scientist during the course of your degree, and it's worth stopping, pausing for thought at this stage in your career and thinking, what is this all about? What's the point? Why am I here? And broadly speaking, scientists are about trying to broaden and deepen our understanding of the world around us. And this process typically starts by observing the world around us, and that could be down a microscope or inside a human body, or it could be literally, you know, outside in the world around us. Observing interesting biological phenomena, asking questions about those phenomena, and then trying to make progress in answering those questions. So the starting point for the scientific method is typically observation and the formulation of questions about patterns that we see in the observations we make. And then we try to find plausible answers to those questions and find support for those answers in the form of data typically. And hopefully as a result of that, we develop a better understanding of the world around us, whether it be how to vaccinate people against COVID, or whether it is about understanding why humans evolved in the first place. All manner of biological questions are tackled in this way. So that seems fairly straightforward, and you think, well, that's kind of universal, that applies to science in general, job done, do I need to know any more? Well, turns out, if you look at history, actually, we get quite a nice guide as to the fact that there are quite fundamentally different ways of making scientific progress, of trying to approach that goal of knowing how the world works. And if we look back to, say, the 17th century, if we look back at Francis Bacon, for example, a famous philosopher, right at the sort of, in the period, in the Enlightenment period, when modern science was beginning to take root, when people were becoming, when they were tackling questions about the natural world in a way that looks something like what we recognise as science. If you ask those people to describe what they were doing, they would describe a scientific process that's quite different from the one that we typically use today, and that I'm going to describe to you in more detail in due course. And in particular, Francis Bacon, for example, would have described the idea of making progress through the process of induction. So collecting evidence, and from that evidence formulating theories. And the more evidence that you could amass in favour of a theory, the more likely that theory was to be true, until you got to such a point that you decided that the theory was proven. And this was making progress through inductive reasoning, as it was called, was deemed to be the best way of making scientific progress. And it sounds kind of intuitively sensible. But there's a really significant problem with this idea of gathering evidence, looking for support for your favourite theory until you've proven that theory to be correct. And on slide there's a couple of different examples of what I'm on about here. The black swan in that photograph may be unfamiliar to you because it's an Australian species. If you were a European ornithologist and you went out in the world and you observed swans, you would pretty rapidly find that swans are white, okay? And every swan you ever saw would be white in your entire life. And probably long before you died, you would have reached the conclusion that swans are white. So you would have, when you first encountered this bird, you would think, oh, that's an interesting large aquatic bird, let's call it a swan. Oh, it's white. And gradually you'd collect evidence that swans are white. You'd start to formulate the theory. You would spot the pattern that swans are generally white, and you'd formulate the theory that maybe they're always white. And maybe if you're a British person, you might travel to the north of Scotland or to somewhere like Slimbridge, and you discover that there are different species of swan. There are actually 3 common species of swan in the UK, or relatively common, and they're all white. So eventually you'd have a weight of evidence in favour of this idea that swans are white. But then you get on a ship and travel to Australia, and as soon as you land, you see some water birds, and hey presto, you see a black swan. So this theory that you thought you had proven is suddenly undermined by new evidence. And this is a really central problem with the idea of inductive reasoning. And to sort of hammer it home, there's often gets told this parable of the turkey, which is why there's a turkey on the slide here. This idea that imagine that you're a turkey and you are comfortable living in the turkey hatchery or whatever it is that you've grown up in, and you're taken to market where a farmer buys you as a young turkey and takes you home. And this is a pretty terrifying experience as a turkey. You don't know what's going on. Who is this person who's just purchased me from the livestock market? Where am I going? You get to the farm that they're taking you to, and you've got your suspicions that this farmer looks like they might be trouble, and that, you know, who knows, maybe you're going to meet a sticky end. And the farmer instead releases you into a lovely pen with a nice hutch for you to, or hutch? I don't know what turkeys sleep in, but anyway, some kind of nice place to stay overnight, and there's bedding and there's food and water, and it seems like actually a really cozy place. And so you settle down, and the day goes by, and evening falls, and you see the door to the farmhouse open. There's a creak, and the light shines out, and it's all a bit spooky, and you're getting a bit scared. And stomp, 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 the farmer comes down to your enclosure, opens the gate, and you think, hang on, there could be trouble here. And what happens is the farmer actually puts down a great big bowl of new juicy food and freshens up your water and gives you some new bedding, and you're like, wow, this is brilliant. And so the farmer goes off, and the next night rolls around, by which time you've scoffed all your food and got a bit fatter, and the same thing happens again. It's night, it's a bit spooky, the door opens, and you think, well, last night this was good news, but I'm not so sure. And the farmer turns up, and sure enough feeds you a lovely meal and gives you bedding and all that kind of stuff. And this goes on night after night, and so soon you get into the swing of things, and by the fourth or fifth night you've formulated this idea that that's what farmers do. They kind of come and look after you and give you like really nice food, and you get fat. And so pretty soon your theory has been confirmed by the weight of evidence, because night after night you've had this wonderful treat, and life is just, you know, absolutely wonderful. And of course, just at the point that you've, as the turkey, formulated the idea that this is what farmers do, they only have turkey's best interests in mind, it's December the 24th, and the door creaks open, and this time the silhouette looks slightly different. There seems to be something in the farmer's hand, and the next thing you know, you've had your head chopped off. So a bit of an extended metaphor for the problem of induction, the idea that simply accumulating evidence until such time as you decide you've got enough to support or prove your theory isn't necessarily an adequate way of making scientific progress. And that has led over the years, since the sort of origins of scientific thinking, to a search for something a little bit more methodical, which might allow us to reach conclusions in a slightly more objective and definitive way. And in the 20th century in particular, philosophers like Karl Popper on screen here, thought deeply about this and came up with a model for the way that science gets done in modern science, called the hypothetico-deductive model. Right, so this is deductive rather than inductive. There's a contrast there, and you'll see how that works. So the idea that Popper, the way that he described how we do science, and it's not that he came up with a new way of doing science, it's more that he described how there was a transition during the 20th century towards a new way of doing science. The idea was that instead of accumulating evidence until such time as you decided you got a workable theory, actually science had flipped that on its head. And the way that we tended to approach science in this hypothetico-deductive model was to develop a theory first of all, and then go out and search for evidence to show whether or not the theory was correct. And so the first step was to observe the world around us and generate theories about what was going on, generate possible answers to the questions that we formulated. And this was a really creative process. It involved, you think of scientists as being boring, conservative people, but actually they are potentially really quite imaginative, creative people, or they should be, in order to come up with interesting explanations, plausible explanations, alternative explanations for what they see is going on in the natural world. And this creative process gives rise, under the sort of hypothetico-deductive model, to big ideas, big theories and models from which we generate much more specific hypotheses about exactly what's happening in each element of a system that we're studying. And we're going to see an example of that in a moment. And this word hypothesis essentially means a statement about the way that you think the world works, a postulation, should we say. And it gets used extensively in science, and it unfortunately, or slightly confusingly, does get used in slightly different ways. Sometimes it's a specific postulation or proposition which takes the form of a prediction. Essentially, this is going to happen, is how a hypothesis sometimes reads. But perhaps more normally, it's a statement of this is how things work, and if that's true, then we can make the following prediction or predictions. So hypothesis and prediction, there's a little bit of a grey area there between the two, but essentially a hypothesis is a statement about what you think is happening, and from it should hopefully arise predictions which you can test. And a crucial thing is that a big idea, a big theory that you might have formulated about how the world works can give rise to actually a whole range of hypotheses. So you can have multiple hypotheses for a single overarching theory. And this is the starting point for the scientific process, this creative idea generating process under the Popperian model. Rather than starting with data in the form of lots of observations of swans on a lake and then allowing those data to give rise to a theory that we feel that we've substantiated. So I think it's easiest to think about all of this in the context of a real world example. So let's look at a real world 17th century problem, which is the plague. And it's amazing how much the science of the plague has progressed over the years. Literally today I found a new paper that's just come out this week about the epidemiology of the plague, about how plague epidemics worked, and we still don't know quite a lot about this phenomenon. Anyway, big issue in the 17th century, killed millions and millions of people. I don't know if you know, but in the 14th century, the plague killed something like a third of the world's population, which kind of makes COVID look like a bit of a storm in a teacup. But come the 17th century, the plague was still around and it was causing major epidemics in the UK, amongst other places, and killing thousands and thousands of people. So this is a big issue, and it's a big natural phenomenon that scientists are naturally going to be wanting to address. They want to know what's going on, where does this plague come from, how do we stop people from dying. The observations that a scientist might make about the natural world are things like this. Well, first of all, that there is an epidemic. Lots of people seem to be getting sick of this sort of identifiable disease. And if you look a little bit more closely, the people who seem to be getting sick are people in the stinkiest, horriblest, dirtiest parts of town, because in a city like London, you'd be seeing huge levels of deprivation amongst a large portion of the population, no sanitation, no sewers, really poor healthcare, if there is any healthcare at all, and poor understanding in the 17th century of, no understanding really of things like microbes, for example. So, but the conditions are not universal. You've also got wealthy people who live kind of on the top of the hill, out of the way of the paupers, and they have relatively good conditions to live in. And it's the people who live in the stinkiest parts of town who seem more likely to get plague and die from it. And these are the sorts of observations that we might make as a good scientist, which then lead us to start to generate some ideas about what's going on now. I've got a question in the chat, which is, can we print out the slides at the end of the lecture? Where will they be? Good question. So yeah, the slides are always available as a separate PowerPoint file on Moodle. So because these slides are being presented to everybody on all the different degrees, they'll be in the Moodle generic content page. So if you scroll down that page, you'll find the different topics that I'll be dealing with, and in each case there'll be links to the recordings, there'll be a link to the slides, and there might be some other activities, quizzes and other things that I chuck in there to help supplement that you're learning, but we'll see how we go with that. And while I'm answering the questions, so prediction is explaining what we think will happen when our hypothesis is tested. So a prediction is what you think will happen when your hypothesis is made real, as it were, when it's tested, so that's right. Okay, so we were talking about this. We've made a bunch of observations about the natural world. And the next thing a scientist does in this hypothetico-deductive model is to formulate a question, the answer to which is going to be one or more hypotheses. So the question we're going to look at here is, what's the cause of this epidemic? Which is an obvious question you might ask as a scientist back in the 17th century. And believe it or not, one very popular idea about what was going on was that something called a miasma was responsible for the plague. In other words, the stinkiness that we could smell, the general stench of human existence, was in some way capable of carrying disease. I mean, it's a kind of natural thought that stinky, horrible, bad air is not good for you. And so it was a natural extension of that idea to say, well, this disease is being carried in the air, maybe by something that we can't see. Maybe the smell itself is itself poisoning people or making them ill. So that's our big idea. It's a pretty big idea that can give rise to actually lots of specific hypotheses. So at this point, we could take that big idea and run with it and generate some specific hypotheses that we can then test. So here's one. Plague is carried in smelly air. Okay, that's one component of that big theory. So the plague is part of the miasma. People catch the plague by inhaling the miasma, by smelling the dirty air, the nasty air. That's how they catch the plague. And another hypothesis is that the transmission of the plague could be stopped, therefore, with nice smells. If we had some air freshener to hand, we could freshen up the air, make it nicer, and somehow that might stop people getting the plague. So these are all potentially testable hypotheses. And at this point, we would deduce predictions from these hypotheses, which would be testable by collecting data. So this is the deductive part of the process. We're deducing a prediction from a hypothesis, which we are then going to test. And we're going to look in my example at hypothesis 3, which is about the transmission of plague being stopped by nice smells. And we're going to do an experiment. Obviously, this is not a real experiment, but we're going to imagine it happened. Based on the prediction from this hypothesis that if you intervene and give people sweet-smelling flowers to freshen up the air around them, you might be able to stop them catching plague. That's a prediction which follows on from that hypothesis. So we've deduced this prediction, and now we want to test it. So how do we test a prediction? There's, broadly speaking, two ways. We could either go out into the natural world and observe what's going on. So we could observe places where there are flowers and places where there aren't, and we could see whether there's an association with good health. Or we could do an experimental manipulation where we intervene and actually provide some people with flowers and others not, and see if it has an effect. And in general, experiments tell you more about cause and effect because you're manipulating something and looking for a response. And this is something I'm going to talk about quite a lot more in a later lecture. But for the time being, let's imagine we did an experiment. And that experiment would involve getting a whole bunch of people who are at risk of catching the plague and giving half of them, chosen at random, some sweet-smelling flowers which potentially could neutralise the miasma and stop them getting the plague. And the other half of the people, well, unfortunately, they don't get the potentially useful treatment. Instead, we're going to give them the nearest possible equivalent, which would be, I guess, some cloth flowers that you could get made in those days, because we want to be wary of any unintended consequences of the treatment that we're applying. So giving people flowers not only produces nice smells, but of course also looks nice, which maybe cheers them up, and maybe that will stop them getting plague. So we want to make the distinction, the differentiation between the two groups in our experiment as clear-cut as possible, so that the only thing that's different is one group has the nice smells and one doesn't. So we're going to give the control group fake flowers, which look nice but don't smell. And then we're going to wait and we're going to see what happens. We're going to look to see how many people get infected. And if this all sounds horribly familiar and kind of like dicing with death, well, it's pretty similar to what's going on with COVID. You know, we've got people who are developing vaccines or treatments for COVID. They think it might work. We haven't got a lot of time. We've got people at risk of dying here and now. And so we have to start doing these kinds of experiments to find out whether people will indeed be saved. So at this point, you might think, right, yeah, so we're going to collect some data and then we're going to hope to prove that our hypothesis about the miasma being stopped by nice smells is correct. And this is probably the most important insight that Karl Popper had about the way that science was going in the 20th century. Actually, that's not true. We generally Popper reasoned, felt as conservative scientists that it was not possible to prove a hypothesis to be true because of the problems with inductive reasoning. For example, you know, we saw the example of the turkey and the black swans. It's always possible to find some new evidence which changes your view of a hypothesis. So the idea that when you've got good evidence, suddenly the hypothesis is proven to be true was something that Karl Popper characterised as being inherently at odds with modern science. It's not conservative enough. It smacks of jumping to conclusions or not being open-minded about the idea that new data might arise. Hypotheses can always be falsified was the sort of the theory that Popper espoused. And instead, as scientists, we view ourselves as these kind of truth seekers. We're looking for the truth, but it always remains out of reach, in the sense that the best we can do is come up with a good hypothesis which has not yet failed, but we always expect that it's possible that it will fail. And this part of how modern scientists approach increasing knowledge is often overlooked by scientists themselves, but also the public and the media. And it can generate all kinds of difficulties when scientists talk to the media. If you've heard scientists talking to the public or on TV or radio, you'll see that they're inherently very conservative. They're very reluctant to be dragged into saying, yes, this is proven to be true. And it's because of this underlying knowledge that in the past and in the future, scientists will end up being proven to be wrong when new data come to light. And the kind of classic example with this, where this causes a problem, I think, is in climate change, because climate change is happening, right? I mean, as a scientist, I'm pretty convinced of that, and I would say it's, despite all the shenanigans with COVID and Donald Trump and everything else, this is the biggie. Climate change is the thing that threatens all of our existence. It's the thing we've got to do something about now. But I'm a conservative scientist. So if you asked me, is it proven that climate change is happening as a result of human activity, the release of CO2 into the atmosphere, I'd say, well, we don't ever prove anything to be true. We think that's what's happening. I'm very confident it's what's happening, but I'm always going to allow for the possibility that the rise in temperature we've seen of late is caused by something else. And at that point, the media scream, oh, climate change denier, or worse still, they take your quote out of context and they use it as, you know, they say, Boffin from University of Nottingham says, actually, we don't think climate change is happening. And of course, that's not what I said. I said, I don't think it's proven yet. And that's because it will never be proven, because nothing is ever proven in science. So with climate change, what you'd really want to be able to do is an experiment where you manipulate carbon dioxide levels at a planetary level and see what happens to temperature to try to get supporting evidence for your hypothesis. But that's not possible with climate change. So it's one of the reasons we're stuck with observing a single planet and looking at what happens and modelling and predicting what might happen. And so we're particularly conservative when talking about climate change. And I think that was at the heart of the problem with climate change denial, because it gives climate change deniers a little chink of light, something to get their crowbar into, and to pretend that there's a great uncertainty in the scientific community about whether it's really happening. It is really happening, we think. We're pretty confident, but we're never going to say it's proven to be true. Instead, what we do is we look for evidence to reject bad ideas. And of course, the good idea that is climate change, the good hypothesis, I mean, it's not a good thing, but it's a good scientific theory, and it has stood the test of 1,000 tests with data, because it's not a dodgy hypothesis. And so we have failed to refute it, to reject it. But this is the destructive part of the modern scientific process that Karl Popper described, that what we do with data from experiments and observations is attempt to knock down, to falsify poor hypotheses, rather than prove good hypotheses to be true. So instead we end up with a kind of Darwinian process, the survival of the fittest. We have competing ideas, we throw data at them, and the ones which survive being subjected to this kind of ordeal are the good ones, the ones that it's difficult to find any evidence that they're wrong. And climate change is exactly one of those good ideas. But it doesn't mean that something might come along in the future, might not come along in the future and show us to be wrong after all. So science is a kind of evolutionary process, but it goes from this creative brainstorming stage to this destructive survival of the fittest stage. Right, so going back to our plague example, at this stage of the process, we would gather our data from our patients or our volunteers who'd had the flower treatments, and we would discover that just as many of them who'd had the flowers died as those that had had the fake flowers. And so there wasn't seemingly any evidence here to support our hypothesis about the miasma. So we might reject that hypothesis. The transmission of plague can't be stopped by nice smells after all. Oh dear, back to the drawing board. So at this point, we would go back to our theoretical understanding of the system, and we would try to generate alternative hypotheses which might be better and might stand the test of experimental testing. So through this process of rejecting poor hypotheses and generating new ideas, sort of alternating constructive and destructive process, we make scientific progress. We get closer to the truth in the knowledge that we'll never be sure we've got to the actual truth, which is a bit depressing in some ways, but that's how it goes. So in our case, with the plague example, what we might do is design a new experiment to test a slightly different version of a slightly different hypothesis derived from the same theory about miasma. So we might say, well, okay, maybe you can't kind of neutralise plague just with nice smells. What you've got to do is get rid of the miasma in the first place. So let's run a program where we give everybody instructions to leave their windows open at night when they sleep to try and clear the bad air from their buildings, and maybe this will reduce the prevalence of plague. And we could go on like this ad infinitum. What would happen is we'd keep generating hypotheses about miasma, and we'd test it and we'd find that it was wrong. And so we'd keep rejecting crappy hypotheses about miasma causing plague because actually it doesn't. And eventually we get to the point of thinking lots of the hypotheses generated from our theory are wrong. Maybe the whole theory is wrong. Maybe plague is not caused by miasma in the first place. And that is, of course, what happened. And indeed, we came up with more compelling hypotheses for what happened, different theories to explain the plague pandemic. And the shift in thinking from one whole set of hypotheses to another is often characterised as what's called a paradigm shift. A paradigm being a whole way of thinking, a whole theoretical structure. And with a paradigm shift, we go back to completely blank piece of paper and start again with the scientific problem and try to work out what on earth's going on. And if you think that sounds a bit unlikely that a sensible scientist would ever come up with a good theory, with an interesting theory that eventually ends up being binned and replaced with a completely different one, you don't have to go much further than Newton and Einstein to realise just how profound paradigm shifts can be in science. So Newtonian physics was an extremely good model for understanding how the physical universe works, developed by Newton, but it took until the early 20th century for Einstein to notice that the cracks that had been beginning to form in that theory as people tested different elements of Newtonian physics reflected a fundamental problem with Newtonian physics, and essentially Newton was wrong. He was kind of nearly right, and Einstein's new ideas about relativity ended up completely replacing the original theories of Newton. Okay, I've got a question here. Is it possible to predict that the hypothesis is wrong when it's tested? Well, I suppose what you're saying there is if you think you've got a dodgy hypothesis before you even start the experiment, can you sort of say, I think this hypothesis is wrong, I'm now going to go and see if that's correct? That's a slightly weird way of thinking. But as we'll see in a minute, what you have is a hypothesis and the opposite of the hypothesis, called a null hypothesis, in operation as a twin pair every time you do an experiment. And so in effect, your question becomes redundant because you always have the two possibilities, equal and opposite possibilities in mind when you do science. Okay, so right, with the miasma theory about plague, we're going to get to a point where we've scrapped the theory, we come up with something new. In the case of plague, we might notice that in these horrible hovels in London, there are loads of rats, and the rats carry fleas, and etc., etc. We might start to put together a new theory. Although, interestingly, the latest evidence suggests, I think, that fleas probably didn't play a very big role in transmitting plague from rats to humans, that instead direct human-to-human transmission was much more important, in the same way that COVID gets transmitted. Right, so this, what I've just described to you is this hypothetico-deductive model of doing science. But in itself, it is really just a model or a theory or an idea about how science happens. Popper was trying to sort of describe the modern model of science and contrast it with different ways of doing science that had emerged in earlier centuries. But it is a reasonably good approximation for what most scientists do nowadays. It looks like a neat linear process. In fact, it's not that neat. You know, sometimes we jump in halfway through. Somebody gives us a hypothesis without us having formulated it ourselves. Sometimes this process even kind of goes a bit backwards. It's not as neat and tidy as it seems, and yet it is a reasonably good model for what's going on in the minds of a scientist, and in terms of the expectations that the public and our peers have of us as scientists. They're expecting us to go through this process. And in fact, for many scientists, they do it without even realizing it's what they're doing. Okay, so as you have seen, a crucial part of this process is the idea of a hypothesis. So that's what I want to focus on for the last part of the lecture, is what is a good hypothesis? Because you're going to need a good hypothesis to do good science. And the answer is, well, a really clear, simple hypothesis is a good hypothesis. That makes a lot of sense. Pretty intuitive, really. I won't say any more about that. If it's a jumbled or difficult to understand hypothesis, clearly it's not going to be very useful. A good hypothesis is normally precise and specific. So let's look at an example. Imagine you're asking the question, why do people with the sickle cell trait rarely catch malaria? So sickle cell trait is this thing where your red blood cells are a funny shape. If you are homozygous for this trait, actually you get really sick with sickle cell anemia. But if you're heterozygous, you've only got one copy of the gene, then actually you're reasonably healthy and it's really difficult for you to catch malaria. So you might not know all of that story. You might simply be looking at people who have this trait and wondering why it is they don't seem to catch malaria or they don't get it as bad. You formulate a hypothesis, which is people with the sickle cell trait are resistant to malaria. Okay, that's a perfectly reasonable hypothesis, you might say. It's simple and clear. Oh, by the way, notice that I've denoted it H1 here, which is the standard way of denoting the hypothesis, or what you're later going to come to call the alternative hypothesis, but we'll worry about that in a moment. So the hypothesis here seems to be clear and simple, but actually it's quite sort of vague or general, because resistance to disease can happen in many different ways. And this hypothesis doesn't say much about the biology of this system. Well, how does sickle cell trait make you resistant? So we can come up with a more specific, precise hypothesis, which actually is much more, it's much easier to imagine how we can test this. How do we test this general hypothesis that sickle cell trait makes you resistant to malaria? Quite difficult to imagine. Whereas if we say the malaria parasite cannot reproduce in red blood cells in people with a sickle cell trait, it's saying something very specific. It's almost formulated in the form of a prediction, which is ready-made to go into an experiment. So we can go and get blood samples from people with sickle cell trait and compare them with people who haven't got the trait, and we can grow the malaria parasite in the lab and look and see what happens. So it's much more testable because it's precise and specific. A good hypothesis is also plausible, because not all hypotheses that you could possibly think of in your creative explosion of ideas are going to be very plausible. And if it's not very plausible, then it's probably not one of the ones that you want to invest a lot of effort in straight up. Maybe if you run out of any other ideas, fine. But so if you take the example of why pesticide exposure sometimes appears to raise the risk of Parkinson's disease, this seems to be true in some circumstances. You might have the hypothesis, well, okay, patients who have Parkinson's are beginning to develop Parkinson's disease, so they have incipient Parkinson's disease, they are really good gardeners. Something about having Parkinson's disease makes you a good gardener, and so you end up being exposed to lots of pesticide. And so that's the explanation for why there's an association between pesticides and Parkinson's disease. It's not very realistic, is it? I mean, it could be true that somehow having Parkinson's makes you more likely to want to garden or something, but it seems pretty unlikely given what we know about the biology of the human body. So perhaps a more realistic hypothesis, this is one that I've pulled from the literature, is that pesticides, particularly this thing called paraquat, which is one example of a pesticide, causes lesions in a particular part of the brain. and that could be the explanation for the association between pesticides and Parkinson's. And that was the hypothesis that was tested in the bit of the literature that I found when I was looking at this particular idea. So I've got a question here, which is, what would an example of a prediction for it then, since the hypothesis is so specific? Okay, yes. So I think the question is getting at the fact that if you formulate a really nice specific hypothesis, it almost sounds like a prediction. Well, that's fine. If the hypothesis really is basically a prediction, then you don't need a separate prediction. That's what I was saying. There's a bit of a grey area between the definition of a hypothesis and of a prediction. The two sort of can merge into one another. But as long as you have a prediction in terms of what the data will look like if the hypothesis is true, then you have a testable hypothesis. Okay, final thing I want to say is, which is sort of obvious from what I've been saying, that a good hypothesis should be testable. So there are hypotheses you can come up with which are great ideas, but you'll never be able to test them. And one field in which these hypotheses are rife is xenobiology, the biology of alien life. So one question you could have is just, is there life elsewhere in the universe? That might be your overarching question. And you might come up with a hypothesis, well, yeah, animals and plants grow on planets in other solar systems, which is fine, but will we ever have the opportunity to go to other planets in other solar systems and look for things that look like animals and plants? It's pretty hard to imagine. But there is a major scientific project ongoing, has been for decades, called SETI, which is trying to answer this question, whether there's life elsewhere in the universe. And it's testing a hypothesis, which is that intelligent life, so not just any old life, but intelligent life, emits radio waves in some way that could be detectable on Earth, even if it's coming from a different solar system, a different part of the galaxy. And that is a hypothesis which is testable from here on planet Earth, and that's what NASA have been doing for many years. So far, we haven't found them. Okay, so hypotheses are really important, and there are some basic rules there for how to make a good hypothesis. In practice, they are shaped, they evolve during the course of a scientific study, and as I've said, the sort of relationship between prediction and hypothesis is sometimes a bit grey. They can effectively be more or less the same thing. So for example, let's say that we are looking at parasite loads in laboratory mice, and we might hypothesize that being in the lab, because, I don't know, sterile conditions or something about being in the lab means that lab mice tend to be free from parasites. That's fine as a hypothesis, but when we actually develop a specific experiment to test that hypothesis, we might slightly tighten up the hypothesis to be a bit more specific and a bit more testable. It's almost in the form of a prediction, which is that wild and laboratory mouse populations will have different parasite burdens. So I'm introducing the idea that we're going to compare our lab mice with wild mice, and we're going to look for the level of parasite burden, the number of parasites per mouse, and that's the comparison we're going to make. So naturally, this then leads into an experiment where I get a bunch of mice and I start looking at their parasites. So there's this kind of evolutionary relationship between the idea of a hypothesis and a prediction. The last thing I want to say is about the null hypothesis, which you might have heard of before, and it turns out to be crucial in this Popperian view of how we do science. So let me just take a step back and go back to what I was saying, this idea that we can never prove anything to be true. So we're going through this destructive process. All we can ever do is reject a bad hypothesis. So that's all very well, but what happens if you do some science and you can't reject the hypothesis? What have you learned? Because what we've said is you can't prove a hypothesis to be true. So if you fail to reject it, then, well, it's just a hypothesis, but we haven't proven it's true, so we haven't learned anything. That was a sort of central problem in this thinking about the hypothetico-deductive system. And a partial solution to this comes from the idea of having two mutually exclusive hypotheses, which can't both be true. And these are called the null and the alternative hypothesis. And so far, I've been talking about what are effectively alternative hypotheses, H1. But there's a null hypothesis, H0, for every alternative hypothesis, or there should be if you formulated the hypothesis well. So going back to that example that wild and lab mouse populations have different parasite burdens, The null hypothesis for that experiment would be that wild and lab mouse populations do not have different parasite burdens. The two things cannot both be true. Either there is a difference or there isn't a difference. By formulating that pair of hypotheses, and in particular by creating the null hypothesis, we can feel like we're making progress when we knock one of the two options out using refutation by generating data which leads us to reject the hypothesis. And in general, what we target is the null hypothesis. We look for data which suggests the null hypothesis is wrong. So the reason we use the null hypothesis, well, it's quite complicated, but one obvious reason we do is it's a bit easier to formulate the null hypothesis. And there may be one null hypothesis and many alternatives. So for example, when I talked about there being either no difference or there being a difference between these two groups of mice, if there is a difference, that difference could be big, it could be small, it could be positive, it could be negative. There are different kinds of alternative hypotheses. But when there is no difference, the null hypothesis, there's just no difference. There's only one simple null hypothesis. So in general, the null hypothesis is a bit easier to work with, and it sort of reflects our conservative, somewhat negative approach to science that modern scientists have. Yes, we have a kind of creative burst, but then we get all kind of introverted and navel-gazy and aren't willing to commit ourselves to things being proven to be true, which I think actually is fundamentally quite a healthy attitude to have. Yeah, the final thing I want to say is that this is all very well, formulating a good hypothesis, generating some data and using them to test the hypothesis, but it only really works as a way of making progress in science if you have a proper way of testing your hypothesis. And just looking at some data, or doing a trial with your COVID vaccine and talking to some patients and seeing how they feel, isn't a very objective way of evaluating your hypothesis. Humans tend to be subjective in the way that they interpret things. In other words, we are influenced by our own background, our own experience, our own prejudices. What we need is a nice objective way of assessing a hypothesis. And that way, that method, is stats. So the reason I'm going to be talking about stats later on in the course is because it provides the objective method that isn't influenced by our emotions and our prejudices of judging whether or not data support a hypothesis. So that's what we're going to look at in a large chunk of this course. Right, I'm just going to deal with the last question before I go on to talk about next week. This might be a stupid question, but do all scientific projects need laboratory experiment? And can we rely on previous scientists' experiments? That's a fantastic question, and it's going to be the subject, part of it anyway, it's going to be the subject of a later lecture. So the answer is no, we don't always rely on laboratory experiments. We don't always rely on experiments. We sometimes use observations. Sometimes those observations are made by other scientists, like you imply. And we can still draw inferences from those data. Just because they don't come from a controlled experiment doesn't mean they're not useful, as we'll see. And there are different pros and cons to doing a manipulative experiment, particularly in the lab, versus observing the natural world around us. And we're going to look at those pros and cons in a later lecture. Okay, so just want to say now about what happens next. So now you're kind of on your own for a bit, in the sense that there are a bunch of pre-recorded lectures on the generic Moodle page. You'll be signposted to them from your degree-specific page, so you can go through the week-by-week guide in the biology or biochemistry Moodle page, and it will say, right, now go and listen to this pre-recorded lecture. I've put them all up now for this term. There'll be more next term. So in the right week, go to for the next 2 weeks, you're going to be looking at exploring data and experimental design. Watch the pre-recorded lectures. They're broken into 20-minute chunks, roughly speaking, and keep on top of any other degree-specific activities for your core skills module. So each group of students is doing slightly different things, so make sure you read the Moodle page for biology or for biochemistry so that you know what you're doing in each week. But I will be back in live form to help you out every now and again, and to take questions and to give you some quizzes and things. And the first of those sort of top-up sessions, if you like, live sessions, is going to happen in week 9, so a couple of weeks from now, covering the stuff I talked about today and the pre-recorded sessions on exploring data and experimental design. Okay, so Thanks ever so much for bearing with me. Sorry I've taken the full hour, but hopefully you got something out of that, and I will see you in a few weeks.